Okay, we are recording. Okay, then Rachel, can I pass over to you? Are you ready?
Yes,
I
I
was ready
am.
just in case you weren't coming because I wasn't sure.
I am technically
You're
on
here.
holiday, but I would not miss this for the world because I am so excited to introduce our speaker today, Dr. Kate Lines. She is an integral part of the neuroendocrine tumor research community, and she has really been pioneering efforts over the past couple years to bring together the community with a common goal of coming up with better preclinical models to study neuroendocrine cancer. Dr. Lyons organized the first ever Net Models meeting last year, and then she's doing so again this year, which we will all hear more about. So, without further ado, Dr. Kate Lyons, take it away.
Thank you very much. I'll get you to do all my intros. That was very flattering. Thanks. Right, I'm going to hopefully share my screen so you can see the presentation. Let me know if you can you all see that okay? Perfect. I mean, this isn't going to be a very serious presentation. I wanted it to be slightly interactive. So kind of give you a couple of updates on what we're doing, where we are going forward, and that sort of thing. And this is still kind of a work in progress. As Rachel said, this kind of Net Models Consortium only really came to the fore a few years ago. So we're definitely up for any suggestions, comments, or just general points that people think should be considered going forward. So kind of the title of my talk is Improving Net Models through Open Science and Collaboration. Now, these are kind of buzzwords that are used quite a lot, I feel, in the field at the moment, which is why I put them there. But the truth is, this is all about collaborative sort of consortium research with us in the NET community. Before I get into that properly, I thought I would give you a bit of an update on me because I think some of you know me, some of you probably don't. So just kind of my background and where I'm coming at this problem from. So I've actually kind of been in the research field now for scarily longer than I feel I should have been. But basically, back in 2007, I got my biochemistry degree at the University of Liverpool in the UK, where I first kind of started dabbling in cancer and sort of endocrinology, I suppose, because I was looking at breast cancer as a little sort of mini research project. I kind of caught the research bug from there, so ended up doing a PhD. So for those American colleagues, you'll be horrified to know that, yeah, you could go very quickly from your degree to your PhD, and then that only lasts 4 years in the UK. So during my PhD, I moved on to the pancreatic cancer side, but the big PDAC side that people talk about more often than the NETs. But again, that kind of got me then into the pancreatic sphere. So, straight after my PhD, I then started working as a postdoctoral researcher, where I moved to the University of Oxford, where I worked with Raj Thacker, who anyone who works in the net field will probably have heard his name. Um, and kind of the rest, as they say, is history. So, I've been doing kind of net research now since 2012. Um, but I recently made a leap and actually set up my own research lab at Oxford Brookes University. So the biggest sort of note here is that it's the department I work in is purely scientists. So it's very much a science focused lab. So although obviously the work we do is translational, the idea is it's all about the underlying biology and science behind neuroendocrine tumors and really kind of honing in on different aspects of that to see if we can improve generally just our understanding to feed into things like biomarkers, treatments, that kind of thing. It's still very early days. I've only been doing it a year. So I'm slowly amassing a team of people, but at the moment it's predominantly me tearing my hair out and trying to get people to do favors for me. So be warned, anyone here, you've probably already heard from me. Um, it will continue, but I'm getting there. I'm getting there. Of course, I'm also very honoured to be the UK Research Ambassador for the Net Cancer Foundation, which has been a really good sort of collaboration. Obviously, started with Simon, and I'm really pleased to be part of this and to be able to kind of be part of this community and help to bring together all the aspects of research, sort of clinicians, and also the patients. But the main reason I've kind of been invited to talk today is that I am the co-founder and the lead of the Net Models Consortium. So this actually started at a conversation at Endo with the Society for Endocrinology and Dawn Quell, where we said that what they were looking for sort of areas where there were real lacks of information in the sort of endocrine cancer field. And we were kind of approached separately. And then, as many people know, the Society for Endocrinology was kind of helpful and Bioscientifica in the startup of this. But they really encouraged me to try doing something about this because at the time it seemed like quite a daunting prospect. But I think we've made some good progress, and actually, I'm quite optimistic, maybe foolishly, for the future. So I wanted to kind of share that optimism and progress with you today. Before I do that, I would be killed if I didn't put this up. So I already have a PhD student starting in my lab in September. But we have another project that has currently gone live, it literally went live this week for another PhD position. So I'm a co-supervisor on this. But it's being run by Barbara Jennings, who hopefully you will meet at the Net Models Consortium if you come, and she's going to talk about Drosophila. So it's using flies basically as a model of discovering effects, for example, of genetic variants. So there is a menin aspect to this. So if any of you know any colleagues who are interested, I will also say that miraculously, it also pays full international tuition fees. So if you have any international students who are interested in nets and menin specifically and are looking for a PhD, here's the details. The link is there. Please look it up and we would be more than happy to welcome anyone in. Okay, so that's the sales pitch over. So I'm now going to talk to you a bit about the Net Models Consortium. Now, as I said, this is going to be a fairly short presentation, so hopefully we have time for discussion afterwards, but just a few kind of important details. So this Net Models Consortium was founded in 2022 by myself. And also we put together some leads that were kind of subgroup leads to cover the different areas, which I'll talk about going forward. But here are kind of listed the names of the people involved. I think what I really wanted to highlight here is these names hopefully will be familiar to people sort of in the field. First of all, I'd like to say it's an all-female team. So yay for us for putting that together. Not sure how that happened. But also, really importantly, it's very much international. So we have people from Europe, from, well, the UK as well, if we count as Europe anymore, I don't know, and the US. So it really is kind of a transatlantic or European kind of get together, really, to see if we can really address these problems, coming at it from different countries, different issues that we all face, but actually really coming together as a whole community, not little silos in certain institutes or countries that are trying to fight small battles on their own. And the ultimate aim of the thing is to improve the laboratory models we have for net research. And that main way of doing that is to bring together net researchers from around the world, but also to make sure we're better at sharing our work. And as I've put here, the good, the bad, and the ugly. What we've always been very poor at is sharing the things that didn't work or that we realized we did really, really wrong. So, you know, our publications are the highlights possibly of, you know, many, many years of work. And one thing that's kind of where the open science comes in, we kind of want people to be more good at is to actually share with the peers, people in the consortium, not necessarily really publicly, but to say really, at least with the people who it would be highly relevant to make it clear so that people don't keep making the same mistakes. Now, one thing I would like to say before I go on to this is the main effort here is to improve net models. That's not because all the research we've done to date is completely rubbish and we've made no progress. So it's kind of, you know, the fact that we've got research done is why we want more. And the issue we've had in the past is that a lot of the models that exist are substandard, and this meeting addressed some of that. But a lot of good work has been done to date, and as we found out more information, it's highlighted maybe some of the issues we have. So I don't want people to think by any stretch that we're setting up this consortium to kind of say that all the work that has been done prior is irrelevant or wrong or, you know, is not up to a good enough standard. That is not true. There is a lot of good research that's been done, but the truth is to get a lot of the translational, particularly so the more clinically relevant work coming through just much quicker. we do need to improve our models. Also considering the diversity of nets, the number of organs, tissues that are involved. We're beginning now to see the nuances between different, genetics, different cell types, different tissues of origin. And it's only through the research that's been done that we've highlighted that these things that we now need. So yeah, as I said, this isn't to say that everything we've done to date is not useful. It very much is. But we want to make it even better. And we want to really start driving forward and ramp up the amount of information, the speed at which we can get it, and then obviously the improvements we can make for NET patients kind of across the board. So our starting point for doing that was a bit of a labour of love, was to do the first ever NET Models Consortium meeting, which we called the NET Models meeting, that was held in 2024. Now, the Society for Endocrinology and their publishing company, Bioscientifica, were kind of integral to this startup. And as I said, we're involved in the initial conversations. But basically, yes, we managed to organise a meeting that I'll tell you the details of going forward. But the best thing is we managed to publish a paper, which shows you here. And the whole point of it was to show the current state of neuroendocrine tumour research models and our future aspirations. So I said the word models a lot. Now, I know some of you will know what I mean by that, but some of you might not. And John, don't tell me off that I haven't put mathematics in here. I haven't forgotten. It will come in. But from a lab-based perspective, the models that we've mainly talked about and that we use at the moment kind of fall into three categories. So those are in vitro, ex vivo, and in vivo. And those are actually the subgroup leads I mentioned. We had two people that were kind of spearheading each of these different sort of groups. So for those of you that don't know, in vitro basically means we're referring to cells that we grow in plates in dishes in the laboratory. They're single cells on their own that live in little flasks, like is shown at the top here. They just grow in a single layer, most of the time on a flask, and they were probably, most of them, harvested from a patient or a mouse or any other sort of organ we might be interested in. And then they've been cultured, so grown basically for many, many, many years. They're very easy to grow and we culture them for a long time. So we'll come into the sort of caveats of these going forward, but that's just to say they're single cells that are very reproducible but are not closely related to that first tumour that we would have got from the patients. Ex vivo refers to things like organoids, and these are basically made from tissue that is harvested from patients during surgery and are cultured in a very different way, still similarly, but in plates, maybe not flasks. But they much more represent kind of the tissue state that you would see of the tumours actually in the person, which is why here I've kind of just put a picture here that's showing some sort of more tissue related things. So they're kind of like a middleman really between having like a whole tumour grown in a dish and these single cells that we have. And then finally is in vivo. Now, the biggest in vivo we tend to use is a mouse model. And we have those obviously for NETs, particularly a lot of them related to MEN1. That's not obviously exclusive. Any whole organism would count as in vivo, from a fly, a fish, rodents, so rats, mice, even things like ferrets, dogs, all of those can count. But mice are the ones that we tend to use most often. And of course, in those, you've obviously got the tumour in a living organism to look at. So I slightly alluded to this, but just to go into it in a bit more detail. So why do we need NET models? Well, basically, they're important for us to be able to study different elements of the human disease. And these include things like changes in biological pathways in the tumours compared to normal cells. So we can understand what's causing these cells to keep, growing, dividing, not dying like a normal cell will. Kind of almost in line with that is identifying new drug targets. So obviously by understanding the biology, we can work out how we can tweak that to find molecules that would be particularly easily targetable by new drugs we can design or repurposing drugs that are already used. And then finally, testing the effect of new drugs to see whether they are likely to work or not in patients. I mean, that's a very kind of simplified list, but that's just some general concepts as to what we might need them for. Um, and generally, we actually need different types of models for each of these different things. So I think we're not in a world where we're going to have a model that is going to cover all of these bases perfectly. So I think what we need to remember is that we need models that suit the thing we're looking at, and not all models will be suitable for what your particular research focus is. And actually, lots of us might need to use different models in our research simultaneously to really get the answer that we're looking for. Um, so why and how would we use each of these models that I've described? So um, there's pros and cons really of each of the models and things that they're more sort of closely useful for, I suppose. So first of all, just kind of talking about cell lines. So as I said, cell lines we've grown for decades. They are really easy to grow. Most of them grow like weeds, and that means also they're very easy to modify. So, for example, we can change the expression of certain genes and proteins in very sort of well established methods because they keep growing and dividing. It makes that quite easy. Um, Therefore, they could be grown into large numbers really, really quickly. So they're quite good for evaluating changes in biological pathways or performing large experiments because obviously you can have them en masse and you can look at very specific things and make specific alterations to look at a particular biological pathway. So, for example, you could silence the expression or completely remove the expression of something like menin and then see what happens to a specific pathway you're looking at. So say, we want to look at another 3 genes we would expect to be altered after we've lost menin expression. The downside is obviously they're an isolated cell that's been grown for decades, so they are far less representative of human cancer cell lines. Also, as we know, cancer's not just one thing, so the NET tumors we look at are not all identical to each other. So, of course, these cell lines are not giving us a sort of holistic view of what is going on in patients, but they're a good starting point. I think for most of us, this would be our starting point, just to give us pointers just to kind of where we need to get to. The organoids I mentioned are more representative of human tumors. So they're good for things like confirming targets or testing drugs to give you a much better idea of whether they would work on a human tumor that you would see kind of in a patient. However, they're technically much more complicated to grow. They also tend to grow for a very limited time frame and they're quite expensive. So you wouldn't be able to grow those en masse to test lots and lots of different things. So you would already have to know kind of what you're looking for. And then finally, animal models. So as I've said, for example, mice, because they're whole body systems, it means that you can look at global effects. So obviously we talk a lot about hormones in the NET field. So obviously you want to maybe see what effect your drugs or just that tumor development in line with its sort of hormone release might have on other tissues, or things like toxicity of a drug. So is it going to be having different effects and actually cause lots of problems that you wouldn't be expecting? However, clearly they're not human. So you can do things like put bits of tumors into mice and grow them, which can also help. But because they're not sort of native in how they've developed, that can still be quite artificial. But of course, the other things you're looking at, the immune response, the general environment, all of those things are obviously not human. So there are going to be some differences. So realistically, I think we probably need all 3 of these areas for all of our research going forward, to really sort of hone in on all the different problems that we've got. But what do we have? So we haven't done research for decades with absolutely nothing. We haven't just been making things up for years, and we do actually have a number of things that are already available. The caveat being maybe not available to everybody. But this was all summarized in that paper we published from the meeting that we had. And actually, we do have over 20 cell lines from different organs, including things like, adrenal glands, pancreas, pituitary, small bowel. So there are cell lines out there that are usable and people have used successfully for quite a long time. There are also so patient-derived organoids. So these are the ones where it's, tissues from patients. Um, Organoids, tumoroids, spheroids. There are specific definitions for each of these things, which I won't sort of bog down with now. But basically, it's the difference between whether they grow long term or short term and sort of how closely related they are to the original tissue. But there are a few of those that exist. For example, lung and intestinal nets that you can use long term and have been used in a number of studies. Um, so I think what you're beginning to maybe see here is that it's kind of tissue specific. So there are some areas where we're doing well and have got a lot of resources available, whereas there are others, for example, the pancreas, where I mainly work, where actually we're lacking quite a lot. So there are certain areas that we maybe need to focus on more than others. We've also got models for over 30 different animals from different species, although, as I've said, mouse is by far the most common. But this table here, so GEM just means genetically modified. So there are genetically modified models, including Drosophila, zebrafish, and rodents. And what this means is that we've particularly changed one gene we know to be involved in NET development, and therefore we've intentionally adapted these different animals so that they will develop the tumours that we want them to. Spontaneous means that they've just spontaneously acquired some mutation that has caused them to get tumours. So that's mainly the rat model. And the dogs, which we'll go into kind of hopefully later on. And then xenograft is what I mentioned, where basically you can take a bit of a tumor or some cell lines from humans and put those into an animal. And that comes with different sort of caveats. For example, quite often you need to use mice that don't have a functioning immune system because otherwise it won't accept it. And so, of course, then that system isn't necessarily as accurate as you would like it to be. But also, why do these need to be improved? Well, as we've kind of gained more information and research has moved on in the NET field over the years, we've kind of noticed some differences in all of the different tumor types that we're seeing. And I think what's become really apparent is that there are lots of different genetic backgrounds. So we've now uncovered a lot of genes that are involved in the development of these different tumors. That there are different stages of disease that we need to consider. So obviously you've got the really aggressive, advanced, sort of more carcinoids, which we're not focusing so much on in this meeting, which is why it's called the NET models meeting, because those are maybe less lacking than some of the others that we want. But especially for early stage tumors, which are the ones that, have less sort of metastasis, less sort of poor prognosis, but grow much more slowly. Those are the ones that we're really struggling. But of course, those would be the ones that are good to understand, because if we can treat things early, then it makes life much easier all round, and particularly will help to improve things like survival rates. I've also learned about all the different hormones that can be produced. So, across the different organs, there are many, many different hormones that can be produced, and we need these to be represented in the models that we've got. Also, there are now inherited disorders. So I've already mentioned genetic backgrounds, but there are some sort of real key drivers that we know about that it would be really good to have models. And actually, we're lacking. So what, for example, is interesting is that a lot of the in vivo models are based around the MEN1 gene, but all of the cell lines we've got are generally not. So, there's a few sort of issues with the models that we've been using, and also in reproducibility of the data that people are getting, as they maybe try different experiments, because they're not all matching in the way that maybe would be useful if they were. But also, apart from the kind of biological reasons, there are also a few limitations of these existing models for researchers across the world. For example, not all researchers can access them because a lot of them need specific facilities. There's issues with sort of ethics in sharing things. And generally, I think we've not been all that good at talking long distances and sharing these things. A lot of them can also be very technically challenging. So it's not something you can just get from someone and say, oh, I'll start doing that tomorrow. A lot of them take a lot of time and effort to establish properly, and also they can be quite expensive and time consuming. So again, funding the amount of money each lab's got to do stuff can also be problematic. Um, and this is kind of why the word collaboration and open science is coming to the fore. Um, but again, what are the major barriers to developing new models? Why have we not got lots and lots of them already in place? Um, well, there are a few factors. One of the big ones is access to human samples. So I think a lot of us have said that, we would love to try all these different things, but the truth is, unless you have a real plethora of samples coming to you, it's really hard. You might only have a few coming in quite slowly, which means that you can't test lots of different things, and it becomes hard to kind of come up with like proper consistent protocols for doing the work. Um, Sharing human samples across institutes and countries is nigh on impossible most of the time because of the ethics associated with it, which, is understandable because obviously you shouldn't be able to just access any, patient information, but also it can be quite a hindrance to sharing resources. Um, Ready availability of the technical protocols. Most of us, if we write a protocol in a paper, you wouldn't have a clue to even begin how to actually replicate that in your own lab. We're all terrible for that. In fact, quite often you end up on some endless loop that references you back to 30 papers along the way and you never actually find a protocol full stop. Um, but the biggest hindrance, I think, across all of it, which is very sad and is the biggest hindrance to a lot. And I got asked this question, which I thought was brilliant at a public engagement event, which was how on earth did they manage to develop a COVID vaccine and all these treatments so quickly? And the key thing all of us said was money. They threw money at it, every lab, everywhere. All they were working on was COVID for like a year. And people were funding it right, left, and centre. And that's how you really leap forward. Well, obviously, we're not going to do that. But I think money has to be a big consideration in what we're doing. We can't hide from it. The only way we're going to be able to do this work is if we get the funding to do it. So that's all the problems, but what are we doing about it? None of these are insurmountable. I think, as I said, I'm optimistic that we're already beginning to make some inroads into doing this. And I think the first thing is we're talking about it. For the first time in a long time, there are lots of groups around the world that are actually talking, sharing, and trying to make changes. So no one is hiding and saying, this is my model, I'm publishing this, I don't want anyone else to do it. Everyone has been really open and happy to have conversations. Um, so based on that, we're hoping to host an annual meeting, which is going to be the Net Models Consortium meeting, where everyone can come and present and discuss their models. We want to share or promote the sharing of protocols and resources, at least between the consortium, if not openly. So maybe asking people to deposit more detailed protocols in open resources so that they're much more searchable and accessible for everyone to see. Also, I think transparency in publications. So I think most of us, as I said, we publish the highlights, but it would be good if people were to publish any unexpected findings, not necessarily negative findings, but definitely say, for example, you found an unexpected genetic change in your cell line or your model that you used. I think it would be quite important for us to know about it, even if it's put somewhere in the supplementary. People who are interested would be able to find it, and it may well be very useful to all of us. But the big one is to kind of foster new projects and grant applications. And that's the main way. That the models meeting is to get everyone in a room together to discuss things so that we can start new ideas and then putting in some real, proper projects to try and start pulling in some money. And if enough of us do that, I'm confident that, we can make some real changes. So, as I've mentioned this word a lot, I think collaboration is the key to really kind of driving this forward. So that leads me on to the next point. And The one big thing you'll notice here is that the NET Models meeting in 2025 is planned, is going to go ahead in Oxford. But it no longer has the Society for Endocrinology badge to it. Instead, the Net Cancer Foundation is going to be heavily involved in organising it instead. So we are partnering with them to move it forward, and hopefully we'll be doing that for many years to come. Which is good because it gives us the freedom without the kind of pressure of a publishing company who obviously want to meet sort of profit, well, maybe not necessarily profit, but definitely have a financial interest in what we're doing, and potentially, have control over things like the program, the costs, etc. So it now means that it's all kind of in our hands. So what we have decided is that it will take place in Oxford, the 11th to the 12th of September. It will be at Headington Hill Hall, which is this place here in the middle, which is actually attached to Brookes University, where I work. And we have two speakers that are organised. So we have Cynthia Anadedu, who some of you may well have already met, and Florin Bouchan from London. Just to give you a bit of background on each of those. So Cynthia is a professor of stem cell biology, so a basic research scientist at King's College London. She has worked on endocrine stem cells for many, many years now and is particularly interested in the pituitary and the adrenal glands, and has come up with some really, really interesting work and actually some good models that we can use for this. So those of you that don't know about stem cells, it's kind of the idea is similar to, when you're an embryo and you're developing, you have key cells at the beginning that can turn into many different things and ultimately define the different cell types in each of your organs. Same sort of concept in cancer, that you're going to have these cells that are kind of able to turn into many different things and can be really problematic because they can evade lots of the normal systems like your immune system, which would ordinarily kill them off. She's got a really good reputation. She's on all sorts of committees, including the European Society for Endocrinology, European Congress of Endocrinology. She's co-founder of the European Women in Endocrinology Network. So that's a little advert for that if anyone is interested in looking that up and potentially joining it. She's also won many awards for her research. So I'm hoping she'll give a really good talk. She's also very supportive of the consortium, so hopefully that should be really interesting. And the second one is Florin from the Royal Vet College. So this highlights again the different models. So she's a lecturer in small animal soft tissue surgery, predominantly focusing on dogs. She's one of the few recognised specialists in surgical oncology. So actually, there aren't many of her kind of in the world, so it's good that she wants to get on board. And her main research interest is the identification of new models and methods for improving the diagnosis, personalised treatment, and prognostication of both canine, so dog, and human endocrine cancers. And actually, I'm already working on a project with her with pancreatic insulinomas. So this I think she'll be really good to speak about the work they're doing and also how dogs can be really good models for humans for a lot of reasons, including the fact that they develop these cancers sporadically like we do, and they're exposed to a lot of the environmental issues that we have as humans, because obviously they're living in houses with humans. So they're kind of their own little patients, rather than needing to be a sort of, a created model that is not necessarily representative of maybe what the heterogeneity you would see in the human population. So just sort of finally, the details about the meeting in 2025. We have a cap on 50 net researchers that we want to bring together from around the world. The big point to hammer home here is the meeting is going to be free for people to attend. There will be no registration cost. You will have to cover your travel. We can't do that. But there will be zero registration cost thanks to the Net Cancer Foundation, who are helping us cover the associated costs, including food. So you will get fed whilst you're there as well. But because we're capped on numbers, we're saying that there's an opportunity for every single research group who attends to present their work. We actively encourage that. In fact, if you don't want to present your work, you're less likely to be able to attend the meeting, because the point is this is open and collaborative and everyone should be sharing. We don't want bystanders just coming and listening and not getting involved in the discussions. We're also going to have another workshop type session like we had previously, where we're going to discuss the ongoing issues and how they can be addressed. Similar to all those things I outlined in this presentation here, all of those were outcomes from the workshops and discussions we had at the meeting last year. Not my opinion, that was the opinion of the 30 researchers that were in the room there at that meeting. Also, like we did last year, all attendees will be included on a paper that is published from the meeting. So that's really beginning to highlight the people that are involved, the different research areas, but also your name. So if people want to contact you going forward, then everyone knows who was involved. So this is kind of my final slide, just to say that the registration opens on the 1st of April, so imminently. So have a look for it. This is the website address, but most of you here will know the NETChatter Foundation. So if you go on their website, it will be obvious. So all the other details will be on there, including the full details of things like the program. So please go on there, have a look, and we hope to see as many of you there as possible. So thank you.
Thank you so much, Kate. I am so excited. I'm ready to be in Oxford now. Does anyone have any questions or comments about the upcoming meeting or the group as a whole? I hope this means we'll see everybody there. Oh, John.
Yeah, I have a quick question. Obviously, there'll be some patient presence there, right? Because the Net Cancer Foundation is sponsoring it. That's awesome. I was wondering
Absolutely.
if
And recommended how we can better integrate the patients as well.
yeah, that's kind of what I was wondering. It's so obvious and so wonderful how everybody's going to come together to talk about it from, basic and translational, and it sounds like even getting on a clinical perspective, right? So I was curious about how the how patient stuff might be integrated in there. What considerations you've given into it. I don't need a specific answer, man. I'm raising the point.
Yeah, so we talked about, even having like a patient. We haven't obviously finalized the whole schedule yet. We've got, the keynote speaker nailed down and one other speaker, and, encourage people, anyone who registers will submit what they want to talk about. So we've talked about, potentially having a patient speak.
Sure.
And we are kind of going back and forth, and we're open to, whatever anyone thinks to the best way, because obviously patients are, one of the most important aspects of this. And how can we best, especially, the patients that, are so deeply involved in the research, how can we best, utilize the patients and their input and what they contribute to the field? Absolutely.
Yeah, and two things to kind of add to that. One is that we're hoping this will expand going forward. So obviously we started it as a fairly small group and are limited on numbers. If it's successful and we manage to raise funds, because that's the other thing, then obviously we can try and have as many schemes as possible. I mean, I know I've had some discussions with Jo Gray from Amend UK, who is obviously the
I know her.
UK charity. So I think the concept would be going forward whether we could start involving those groups as well, so that we almost have like a sort of Net Cancer Foundation, Amend, any others that would be interested kind of coming along and maybe having mini sessions that they can present, and any patients. The other thing, I think one of the easiest ways to tap into the patients is via the clinicians. And I've already been approached by a few clinicians in the UK who don't do research, who are purely clinical based. And they've already invited me to go to the UK NETS meeting in December. That's a very clinical meeting, to present the work from this meeting, the Net Models meeting, to see how they can potentially integrate and help. So I think going forward, next year, probably, we would like to have a more clinical and patient orientated section. I've also been in conversation with one of the nurses in Oxford, who's doing her PhD where I work. Um, and I think we'd be quite keen to get some of those nurses in, especially those, because in the UK, I don't know whether it's the same in other places, there's a drive at the moment where a lot of nurses are getting like master's degrees and PhDs. And Brookes has a big nursing school, which is how I've come across some of those. So a lot of those are very interested in research projects and can also help bridge that gap because they are the ones, no offense to all the clinicians who do see their patients, but the nurses tend to be the ones that hang around them a lot more and maybe are more trusted, have more regular communication, and often do a lot of the consent anyway, in the UK at least, for things. So we're hoping if we can tap into that, that could help on both the access to samples that we need as scientists, but also working out how we can translate what we're doing back to the patients and find out how they want to help. And I think also to kind of encourage the patients and educate them on when a clinician or a nurse or someone comes up to you and says, would you be willing to give your samples for research? That we kind of help to explain what that means. And hopefully then it becomes less scary and daunting when you're going into surgery or whatever and someone shoves a consent form in your face that's probably got a lot of scary words on it, and you don't really know what's happening. So there's definitely a lot of dialogue that we want to start having, and anyone who's got suggestions, we would be very, very happy. In fact, if we could have one of the no hints or anything, sort of group leads to be a patient, that would be very, very helpful, I think, to all of us to help guide us on that front. If only we knew one who was interested.
Right. If only.
I wish we knew a patient that did a lot of modeling
Can I
here.
nominate John?
I'm always happy to help. And you all know this, always happy to help at whatever level is helpful. So happy to talk.
Plus, we need some more Y chromosomes on the organising list because I'm not sure what we've done. Otherwise, we'll get a reputation.
Well, I did notice a lack of diversity in that regard.
I was going to say we're really, really poor on the diversity front over here. I'm sorry.
Okay, great. Thanks for responding to that question. And sounds like you've got a great start.
Yeah, I just want to confirm, so for those presentations for September, they are not necessarily have to be on model, right? We can have like a presentation using this model on new therapeutics or some pathways.
Yeah, yes. I mean, so for the registration, we're going to ask for a research summary from people, and that can be on whatever you like. I mean, it would be really boring if all of us just presented the caveats of, and the, things we've done wrong with our models. So for the last meeting, generally people did present, experiments they've done with the models they've got, and maybe in that sort of some of the caveats, but generally, yeah, it's data driven because that's what we're interested in as well. And it can be published or unpublished. I think some people were scared to talk too much about unpublished data, especially not knowing the other people. But hopefully, as this meeting goes forward, we'll kind of hopefully develop an element of trust between everyone. Because, if someone scoops someone or does something untoward, if we've got the entire net community at this, you're not going to get very far in your research going forward because we'll all review your grants and your publications. So, word of warning if you misbehave. No.
I just want to add that, Kate, that you mentioned about the element of trust, and I think what the Net Models meeting has created is really this environment of trust too, that we've come together, and now we're getting to know each other much more, and also other people in the field. And I think really this element of trust in the field is really a good thing for the neuroendocrine tumor community, and then and opening doors for collaboration. And so, yeah, really congrats for bringing all of us together. And it's really, I think moving forward, we'll just be expanding, and it's a great thing.
Thanks. So I have to say, everyone's been great on this, and this wouldn't have happened without the support of all the people in the community, because the fact that people are interested and willing to help and put themselves out there. And as you said, I think it is really good that we're getting to know each other, whereas maybe before we didn't. And I think we're learning, all of us are really busy. We don't have time to be fighting each other to get publications and to get different models. And it's far easier if we just work together to move things forward.
Yeah, and like you mentioned, it's really expensive and some of these samples are really rare. So I think that's why it's like just coming together. This movement of coming together is really making a huge difference. So it's good for everybody, and the momentum just feels wonderful.
And I think also we haven't talked so much about paperwork, but I think what we're also learning as a group is how we can, you know, make things move forward, like how we can get cells transferred from different labs, different people, steps we need to take to appease the different, organizations, etc. So even the more we talk, the more easy that becomes. If we have, ways of just kind of getting past all the red tape, then that is helping all of us quite a lot.
All right. Well, if there's nothing else, we can go ahead and wrap up. Like Kate said, the information will be added to the website on April 1st for registration. So please check it out.
And thanks to Rachel for that.
If anyone has any problems with the website, let me know.
Yes, I should add that Rachel has been very integral in this as well and has done a
Right.
lot of the work. So any of these figures I have presented for the models meeting in 2025 were nothing to do with me or the website. So
When I
I have
was
said big
little,
thank you to Rachel for that.
when I was little, my first dream was to be a graphic designer, and that didn't work out. So I became a cancer researcher. But I'm trying my best. I don't have any formal training, and some of the graphics might look a little weird, but
No, we wouldn't survive without you, Rachel. I mean, I know nothing about designing websites, so you are the best.
thank you guys.
Definitely.
Anyway, so if we're going to wrap up, thank you, Kate. That was amazing. And hopefully, you're all as excited as we are for this Net Models meeting. Any questions, then just send us an email. And as Rachel said, and Kate said, 1st of April, then applications will be open, which is next Tuesday. I think we're doing till the 30th. So from the 1st to the 30th of April. So applications open for the whole of April. And then we will then, okay, well, I won't, but everyone else will get together and discuss what has been put forward and what will be the most useful, and we will go from there. But all very exciting. So just to let you know, next meeting is the 24th of April, and we are back to our normal time of our 6 p.m. I don't know what time that will be there because our clocks go forward on Sunday. So I'm assuming your clocks would have gone forward by then, would they? Yeah.
They've
Okay.
already gone forward.
Oh, they've already gone forward. Right.
I know, it's so confusing.
Right, okay. So yeah, back to normal time. So whatever time that may be where you are. But thank you very much, everyone, for coming, and we will see you all next month. Thank you. Thanks a lot. Thanks. Bye.